we are going to talk about the digestive system, which is Unit 8. This
And with her new teeth, she can take on more solid foods. Digestion starts in the mouth. Teeth grind up the food. Then, special glands under the tongue pump out saliva to help break down and lubricate the food on its 12-hour, 13-foot journey through the gut. It'll pass from the stomach into the coils of the small intestine before finally passing into the large intestine. Waves of contracting muscle keep the food moving, a process called peristalsis. These contractions are so powerful, we can even eat upside down. For the first time, a new camera shows a high-definition view of how food travels through our bodies and into our stomachs. Food enters the stomach through a hole at the top. The stomach is a bag of muscle that churns, squashes, and squeezes food into liquid. At the same time, acids break the food down. The stomach walls protect themselves with a lining of mucus. Without it, the acids could digest parts of the stomach itself, causing stomach ulcers. About an hour later, the stomach squeezes the broken down food out through a tiny hole called the pyloric sphincter. The food enters the small intestine, an 11-foot coil of tube where we absorb most of the nutrients. The interior wall of the small intestine is lined with millions of microscopic projections called villi. These increase the surface area of the gut, making it easier to absorb nutrients. First, the pancreas pumps out a juice that neutralizes stomach acid. Then bile from the liver breaks down the fats into tiny droplets. Smaller droplets are easier for the intestine to absorb. After an hour and a half, the small intestine has absorbed most of the nutrients from the food. It's time for what remains to move on. It enters the large intestine through this, the ileocecal sphincter, a valve that keeps our food from going back where it came from. What's left is a mix of waste food and dead cells from the walls of the gut. The large intestine's main job is to extract water from it. Lots of bacteria live here too, but it isn't because of an infection. We actually need them. They produce enzymes that break down complex carbohydrates in our food, carbohydrates we couldn't otherwise digest. Finally, after about 12 hours, we expel what's left of our first meal.
So that kind of gave you an idea of what we're going to be talking about today, the digestive system. And now in the digestive system, the organs within that system can fall into two groups. The first is the alimentary canal, and this is a continuous muscular tube. And this is going to include a few of the items on here. Of course, the alimentary is going to be the mouth or that oral cavity. Next you're going to see is the pharynx, the esophagus, the stomach, the small intestines, which you can see subdivided there. And we're going to talk more about those later, and the large intestines. And basically the main function of that area is to digest and absorb food. So just think, it's that one that follows through that tubing all the way around. The next is basically the accessory digestive organs. Those are, it's not here, but it's the teeth, right? The tongue. There are salivary glands that are located. So all of those are in the mouth. You also have the gallbladder. You have the liver. You have the pancreas. And basically, all of these guys are there to aid in mechanical digestion, mechanical breakdown, and they all help to either break things down mechanically, or they help to produce specialized secretions. And these secretions aid in digestion, right? And so all the organs within the body can fall into one of these two groups. Now, there's three key concepts that help to regulate GI activity. The first is the digestive activity is provoked by a range of mechanical and chemical stimuli. So when we're talking about this, you basically have receptors that are located in the GI tract organs. So those are one way that it can basically determine if digestion needs to happen. Another way that mechanical or chemical stimuli can be picked up is it can happen, or you can get digestive activity happening because of things like stretch or changes in osmolarity or pH or presence of different molecules. So all of those things can help to provoke digestive activity. Now, the next key concept is that the effectors of digestive activity are basically smooth muscle and glands. So when stimulated, the receptors that we were just talking about can initiate reflexes that actually stimulate smooth muscles. And so those smooth muscles are there to help mix and move the content of those that's within that alimentary canal. Now, at the same time, reflexes can also help to activate or inhibit digestive glands. And these glands help to secrete digestive juices or hormones. And the third that we have is the third concept that we're going to hit on is that you'll have neurons, both intrinsic and extrinsic, and hormones that help to create or help to control digestive activity. So the intrinsic controls, these are short reflexes, and we're going to talk about those being regulated by the enteric nervous system, which is within the actual digestive system itself. And then when we're talking about extrinsic, these are going to be longer reflexes, and those are going to be autonomic. And we're going to go into detail about those. When we're talking about hormonal control, hormones from cells in the stomach or small intestines can actually target cells in the same or different organs to secrete contact. And we're going to talk a little bit more about how the hormonal controls work. So there's four main functions of the digestive system itself. The first is to take in food. Obviously, we do that through the entry of the oral canal. Next, what we're going to do, or the digestive system is going to do, is it's going to break it down into nutrient molecules. Once we break down those nutrient molecules, we want it to get into the body. So we're going to absorb those molecules into the bloodstream. That's our highway to get throughout our system. And anything that we don't need, obviously, we're going to rid our body of any indigestible remains. And so those are our 4 main functions of the digestive system. So now let's just, we looked at the digestive system, and I showed the first video to kind of give an overview and so that you could see what it looked like inside of every single organ. I thought that was really neat. But this is going to give us a general overview from start to finish, what's happening in the digestive system. And then what we're going to do is we're going to pull each part separately and talk about basically the process of breaking down food, how food moves through, how we enter it, how mechanical digestion works, how chemical digestion works, and then all the different organs.
Across the whole planet, humans eat on average between 1 and 2.7 kilograms of food a day. That's over 365 kilograms a year per person, and more than 28,800 kilograms over the course of a lifetime. And every last scrap makes its way through the digestive system. Comprised of 10 organs, covering 9 meters, and containing over 20 specialized cell types, this is one of the most diverse and complicated systems in the human body. Its parts continuously work in unison to fulfill a singular task, transforming the raw materials of your food into the nutrients and energy that keep you alive. Spanning the entire length of your torso, the digestive system has 4 main components. First, there's the gastrointestinal tract, a twisting channel that transports your food and has an internal surface area of between 30 and 40 square meters, enough to cover half a badminton court. Second, there's the pancreas, gallbladder, and liver, a trio of organs that break down food using an array of special juices. Third, the body's enzymes, hormones, nerves, and blood all work together to break down food, modulate the digestive process, and deliver its final products. Finally, there's the mesentery, a large stretch of tissue that supports and positions all your digestive organs in the abdomen, enabling them to do their jobs. The digestive process begins before food even hits your tongue. Anticipating a tasty morsel, glands in your mouth start to pump out saliva. We produce about 1.5 liters of this liquid each day. Once inside your mouth, chewing combines with the sloshing saliva to turn food into a moist lump, called the bolus. Enzymes present in the saliva break down any starch. Then your food finds itself at the rim of a 25-centimeter long tube, called the esophagus, down which it must plunge to reach the stomach. Nerves in the surrounding esophageal tissue sense the bolus's presence and trigger peristalsis, a series of defined muscular contractions. That propels the food into the stomach, where it's left at the mercy of the muscular stomach walls, which pound the bolus, breaking it into chunks. Hormones secreted by cells in the lining trigger the release of acids and enzyme-rich juices from the stomach wall that start to dissolve the food and break down its proteins. These hormones also alert the pancreas, liver, and gallbladder to produce digestive juices and transfer bile, a yellowish-green liquid that digests fat, in preparation for the next stage. After 3 hours inside the stomach, the once shapely bolus is now a frothy liquid called chyme, and it's ready to move into the small intestine. The liver sends bile to the gallbladder, which secretes it into the first portion of the small intestine, called the duodenum. Here, it dissolves the fats floating in the slurry of chyme so they can be easily digested by the pancreatic and intestinal juices that have leached onto the scene. These enzyme-rich juices break the fat molecules down into fatty acids and glycerol for easier absorption into the body. The enzymes also carry out the final deconstruction of proteins into amino acids and carbohydrates into glucose. This happens in the small intestine's lower regions, the jejunum and ileum, which are coated in millions of tiny projections called villi. These create a huge surface area to maximize molecule absorption and transference into the bloodstream. The blood takes them on the final leg of their journey to feed the body's organs and tissues. But it's not over quite yet. Leftover fiber, water, and dead cells sloughed off during digestion make it into the large intestine, also known as the colon. The body drains out most of the remaining fluid through the intestinal wall. What's left is a soft mass called stool. The colon squeezes this byproduct into a pouch called the rectum, where nerves sense it expanding and tell the body when it's time to expel the waste. The byproducts of digestion exit through the anus, and the food's long journey, typically lasting between 30 and 40 hours, is finally complete. TED-Ed is a nonprofit.
So that kind of gives an idea of how it works. And so let's go into processing of food. It actually involves 6 essential activities. As you can see, the first is ingestion, and it's right there. So ingestion is the process of actually eating, taking food into the mouth, right? And then number two, whenever we're talking about this, we're actually talking about propulsion is the second. And so this is the propulsion is the actual movement of food. And this happens or involves it going through the alimentary canal. So you can see here, you're going, and it's talking about the movement going through that tubing there. So it's composed of basically two parts that we're going to focus on here is swallowing and peristalsis. And so swallowing is just this idea of making it so that it separates, kind of forms its vacuum to allow it to start going down through the esophagus. And then peristalsis is the major means of moving food or propulsion of food. And basically it's going to involve alternating waves of contraction and relaxation. Now, the third activity or major process that's occurring during the digestive system is mechanical breakdown. You can see that up here. And so mechanical breakdown is going to include in here chewing, mixing food with saliva, churning the food in the stomach, and this process called segmentation. Don't get too concerned about understanding the difference between peristalsis and segmentation because I'm going to hit on this after this. And basically segmentation is local constriction of the intestines that helps to mix food with the digestive juices. And so it's basically showing you this all up here, chewing, churning, segmentation. Now the fourth part is digestion. And so digestion here, I'm going to move this down here so we can just see it a little bit better, is a series of catabolic steps. And we know catabolism is the process of breaking down. And this takes enzymes to do, right? And so steps, and so basically what's happening here is that we're taking complex food and breaking it down into chemical building blocks. Now the fifth part is here, which is absorption. And basically this is the passage of food from the lumen of the GI tract to the blood or lymph. And defecation, which is down here, the sixth part, that's the elimination of indigestible substances. They called it stool. Here, the term they also use is feces. And so this is how food is processed, and these are the 6 essential activities. So what we're going to talk about now is kind of the difference between peristalsis and segmentation and how this affects those activities. So peristalsis, this is when we were talking about propulsion, number 2. This is the main way of moving. So when you chew, the food in your mouth is going to mix up with saliva, and it's going to make this thing called a bolus. And this bolus is kind of this round, packed, entity here, right? And so when you swallow and it starts to go into your esophagus, what will happen is that your esophagus is going to contract. And when it does, it's going to push this guy forward. Now, as you can see at this point, this area that was originally contracted is loose, and the contraction happens farther, pushing it farther down. So this is what's happening. It's basically contracting just farther and farther, and it's going to push it until it gets primarily through the esophagus or here. So the point of peristalsis is primarily for propulsion. There's some mixing that can occur, but it's pushing it through. Now segmentation is different. If you see, this had adjacent segments. So you have this, and then the adjacent segment that's right below it contracts, and then this. But here is different. Here is adjacent segments of the canal will contract. So if you see here, you have the bolus. And what happens is it contracts right here. And so you can see it's pushed down here. So what happens is that bolus, or that gets mixed. Half of it goes here, half of it goes here. And then as you can see, these guys contract here and mixes it back together. So this is basically mostly for mechanical breakdown. You're pushing it. It's kind of like having having some kind of bag and pushing stuff in to kind of mix it in the bag. And you're pushing down on different parts of the bag, and as you do, everything kind of mixes together. That's what segmentation is. And this happens mostly when you're looking at it in things like the small intestines. So that's the difference between peristalsis and segmentation. Now, as we go through the digestive system, it is connected, or basically, the GI tract essentially has 4 different layers, and it's normally surrounded by what's known as the peritoneum. So the peritoneum both has the visceral and the parietal peritoneum. If you remember, when we were talking about serous membranes, this is somewhere on that, right? So the visceral peritoneum actually covers the external surface of most of the digestive organs. The parietal peritoneum lines the body wall of the abdominal pelvic cavity. And you'll remember we learned about that in A&P 1 when we were learning about serous membranes. The peritoneal cavity is located between these guys, and it's filled with serous fluid. So basically it's helping to reduce some of the friction, because as you can tell, just by looking at some of the videos, there is a lot of movement that's happening from the organs of the digestive system. Now the mesentery, which recently they've decided is its own organ, is a double layer of peritoneum. And it extends to the digestive system from the body wall. And so what this is going to do, it's going to allow blood vessels, lymphatics, and nerves to reach the digestive system. It also helps to hold the organs in place, and it stores fat, right? So it has 3 main functions, right? So blood vessels, lymphatics, and nerves to reach the digestive organs, right? 2, keeps organs in place. And 3, stores fat. So that's the mesentery. Now the types of organs you have are the intraperitoneal organs and the retroperitoneal organs. So the intraperitoneal organs are organs that are located within the peritoneum. And the retroperitoneal organs are organs found posterior of the mesentery. And so they're laying against the abdominal wall. So in here, you include mostly like the pancreas, the duodenum, and parts of the large intestines. So let's kind of look at this, at the different parts here. So most digestive organs are the intraperitoneal, and they're suspended from the body wall by a dorsal mesentery, right? And so if you look here, right here is the peritoneal, parietal peritoneum, and then this right here is going to be the visceral part, and then if you look in the middle of all that is the alimentary canal organ. And so most of them are going to fit in this way, and that's the intraperitoneal, if they fit in there. Some intraperitoneal digestive organs are also suspended from the body from the ventral. So this is if they're suspended from the dorsal. This is the ventral. So a perfect example of something like this would be the liver, right? And then some digestive organs are called the retroperitoneal because they have, basically, they've lost their mesentery during development, right? So they're on the outside here. And if you're trying to figure out what the, basically the mesentery looks like, you are going to look at this area that is connecting the small intestines, all that area that you're looking that radiates and kind of keeps it in place. That's what the mesentery, and that's what we're talking about here. Now, when we're looking at the alimentary canal, all digestive organs have the same basic layers, or those layers are called tunics, right? We have the mucosa, we have the submucosa, the muscularis externa, and the serosa. And so we are going to actually look at those basic, and we're going to look at them in the lab as well. So let's look at the basic structure here. And so the most outward layer, the number one that we're going to look at is the mucosa right here. And you can see that mucosa, essentially, this is the innermost layer. And so when we talked about here, we're going from inside the innermost layer to outside here. And so let's start on the innermost layer. So if you see here, here's the lumen. That is basically where all the digestive substance is going to be here. So the innermost layer is that mucosa, and you can see it's made of the epithelium lining that's going to be right here on that inside, right? The next is going to be that lamina propria, and then you're going to see within that the muscularis mucosa. And so when we look at that first layer, this guy right here, it has simple columnar epithelium, if you remember back from the histology section when we're talking about epithelial lining and where certain parts are located, right? It has basically the mucus secreting cells here. And this is basically where you're finding is in the mouth, esophagus, and anus. They are going to be made up of mostly stratified squamous versus simple cuboidal. Again, this area helps. It is the mucus that we have here is there to help protect the digestive organ from enzymes and to eat food passage. And there are a lot of enzymes that can be secreted from there or hormones. The next part here, the lamina propria, this is generally made up of loose areolar connective tissue, and it's basically rich. Let me take this. So the epithelial has the mucus, right, to help to protect it and to help push food across. This guy right here basically has a lot of capillaries in it. And that's important because it's needed for nourishment and absorption. In this area also, you might remember learning about molt before, and that's what you find in there, right? That is that mucosa-associated lymphoid tissue. So this is the area that's going to help defend against microorganisms. And then, of course, this guy right here, the muscularis mucosae, that's going to be important in movement, right? It has smooth muscle here, and so that's going to be what helps to propel with the propulsion of the food across through there. Now, this submucosa area here, this is going to be important because it has a lot of elastic tissue here. So this is a part that helps to regain shape. So the large amount of movement that you see is going to be this muscularis externa. And this is where the brunt of segmentation and peristalsis comes from. Now this one right here, the muscularis mucosa, that is going to be the movement of allow the local movement of the mucosa area there. And basically the serosa is the outer layer. Now, when we talk about this esophagus, we're going to talk about the inventita, and that's what is the serosa is in the esophagus. But this is the basic structure. You can see the plexuses there that'll help with activating digestion. So this is the basic structure of the alimentary canal. So just to review, the innermost of the mucosa, the outermost is the serosa. The mucosa, the epithelium, is going to be where you produce all the mucus, and that's going to help protect it and allow food to move through it, right? The lamina propria is going to be the area that you have malt in, and so essentially that's going to be where nourishment happens and absorption because of the capillaries are located there, and that's going to help defend against microorganisms. And this muscularis mucosa is going to help produce local movement of the mucosa. The submucosa right here is basically going to be the area that has elastic tissue in it, so helps so this organ can regain shape, especially after large meals. This muscularis externa is going to be where segmentation and peristalsis happens. It can even, and you see it has inner circular layers and outer longitudinal areas, so there's two different areas there, and the circular areas are actually will thicken in some areas, and that's where you're going to get sphincters, and we're going to talk more about sphincters later. And the serosa is the outer layer, right? You can actually see it's made up of the visceral peritoneum, and it's a protective layer over the whole thing. So how does, when we're talking about the alimentary canal and we're talking about the digestive system, how is blood supply handled? And so you have both what's known as the splanchnic circulation and the hepatic portal function. And so basically with the when we need the digestive system to actually get blood, we're going to go through the splanchnic circulation. So this is to serve the digestive system. The hepatic portal circulation is a little bit different, right? On here, its function is the drain. So it's going to drain nutrient-rich blood from the digestive organs. And I'm going to put DO for those, right? And it's also there to help deliver blood to the liver so that they can process it. So the blood supply is a little different, right? We need a blood supply to get to the digestive system, to serve it, to give everything that you need. So that's where you have the splenic, the left gastric arteries, and the hepatic arteries giving blood to those organs, along with the inferior and superior mesenteric arteries. So those are all branching off the aorta and it's to serve those digestive organs so that they can have adequate supply of oxygen, adequate supply of nutrients. Now, the hepatic portal circulation is going to be whenever we get done and we're basically trying to put that blood back into circulation. What is it doing? And so in that case, that's where you're going to take that nutrient-rich blood from the digestive organs, and it's going to drain it and deliver it to the liver to process it. So then once everything is removed that needs to be removed from that, it can be recirculated into the body. Now, how do we control the digestive system? And we were talking earlier that we had two basically, internal nervous system, and we have both a localized system and a overall system that helps with it. And so the enteric nervous system is what we're going to talk about first. So basically the GI tract has its own nervous system. It's called the gut brain. It contains more neurons than the spinal cord does. It's made up of enteric nerves that communicate extensively with each other. And so the major one second, guys. So the major nerve supplied to GI tract wall that controls basically motility. Okay. And so the enteric nerves make up the bulk of two main interconnecting intrinsic nerve plexuses. We have both what's called the submucosal nerve plexus, and then we have the myenteric nerve plexus. And so the first regulates glands, and then the smooth muscle in the mucosa. And the other controls the GI tract motility. So now let's look and see where these different plexuses arise within the different layers. If you can see what we talked about earlier, right? The mucosa was here. So that's the epithelium that we talked about was important in making the mucus, right? And then this had the malt, so it protected them. This had the smooth muscle that essentially helped to move mucosa, right? And then you have the submucosa. And the submucosa was important because it helped to basically get the shape back, right? And so this is that first plexus, or that submucosal plexus that we were talking about, that helps to regulate glands and smooth muscle in the mucosa, right? And so it is located in the submucosal area. It's also known as the Meissner's plexus. The Auerbach's plexus, or the myenteric plexus, is located within the muscularis propria. And essentially, remember the thicker circular muscles can essentially be the sphincter muscle, right, or the sphincters that help to separate the two. And this is the area that's responsible for segmentation and peristalsis. So this should make sense that this is located here because this is the plexus that controls GI tract motility, right? So if you think about what each of these plexus does, it should be very kind of forward that these are the areas that they're in, and this is the outside, right? And so this is the protected area here that essentially is comes or is part of the visceral peritoneum. Okay. So that kind of shows you how the general organization of the gastrointestinal tract is and where those different plexuses are that help to basically communicate with that gut brain. And these are part of the intrinsic nerve plexuses, right? So now when we're talking about the enteric nervous system, also participates in both short and long reflex arcs, right? And so there's a whole part that's kind of part of both happens immediately and is quick and in the area, and that's a little bit farther out. And so short reflexes are mediated by the basically enteric nerve plexus, which we just talked about, right? And that's the gut brain. So the short reflexes think it's short, so it's going to respond to stimulus that's actually in the GI tract. Now, basically, the long reflexes can respond to the stimulus inside or outside of the gut. So this can come from the autonomic nervous system. Remember rest or relaxation, fight or flight? So parasympathetic system is going to enhance. So this is rest, relaxation. This is going to be, there's not a bear that's going to be chasing you. So at this point, this parasympathetic system that's coming from the autonomic can help to enhance digestive system to help get those molecules that you need to build things. Sympathetic nervous system is going to be something like a fight or flight, and this is going to inhibit digestive system. You don't really need to digest food while you're running from a bear. And so that's basically how our system regulates digestion. And so here you can see just a nice cartoon of how it works. So if we're looking at an internal stimulus here, this is the lumen of an alimentary canal, right? And so here in the internal GI stimulus, it's going to be something that's going to activate a chemoreceptor, an osmoreceptor, a mechanical receptor, something that's there. And basically the short reflex is going to activate the intrinsic local nerve plexus or the gut brain. And so what happens here is it can activate a smooth muscle or gland. And when that happens, you can get either secretion from the gland to basically start with the chemical digestion, or you're going to get change in the contractile to start with the basically mechanical digestion. Now, the internal stimulus can go also to a long reflex, which means this is going to be something that has to do with the central nervous system here in this long reflex, right? So the visceral afferent is going to go away and talk to the central nervous system. That can also be an external stimulus. So if you see, smell, have any kind of taste or thought of food, that can also talk to the brain, right? And then the autonomic nervous system is going to come in and basically talk and allow for a smooth muscle or gland to also be activated as well, right? And so the long reflex, just think for the most part, it's going to be the autonomic nervous system that's going to either turn it, increase it or decrease it, right? And that's going to be long reflex because it's going to go out of the actual alimentary canal away from the digestive system to decide what's going to happen. Where the short reflexes are going to be basically the gut brain, or one of those intrinsic nerve plexus is going to decide what's going to happen. So now let's talk about all the different organs. So let's start first with the mouth and the associated organs. So first, we're going to talk about the functional anatomy of the digestive system. So mouth is where food is chewed and mixed with enzymes containing saliva. That begins the process of digestion, and swallowing process is initiated. So the associated organs included to this is the mouth, the tongue, the salivary glands, and the teeth. So let's talk about the mouth first. Sure, I'm on the right. And so, ingestion occurs only at the mouth. Okay. It's bound by lips anteriorly, cheeks laterally, a palate superiorly, and tongue inferiorly. And we're going to look at all this, right? The oral orifice is the anterior opening of the digestive system. Walls of the mouth are lined with stratified squamous epithelium, and that's important because these cells are tough, so they resist abrasion. Think about how many times you've bitten on the side and the kind of food that you bring in sometimes is very sharp or pokey, and so every time you bring food in, you're not essentially targeting yourself in the sense that you're going to bleed out through your mouth. So they are pretty resistant to not being broken through. Cells of the gum, hard palate, and part of the tongues are keratinized for extra protection. So they do have keratin, that protein keratin in them, that's going to make them way more robust. And so as you can see, this interior part, or this anterior view of the mouth, right? You have the lower lip and you have the upper lip. And then if you look, you have this kind of separation here that's in the middle. This is what's known as an inferior labial frenulum. And then you have the one on the top that's a superior, right? And then if you look, you essentially have the oral vestibule is that area that's under your tongue that's between your gum line, which is the gingivae, and your lip, right? And then you can see anytime that you have these lines that are connecting them, the frenulums, you can see another one of those underneath, and that's the lingual, right? And that's going to be there kind of helping to connect this bottom area to your actual tongue, right? And you can see these areas right here are the sublingual folds that are going to have the sublingual ducts. And then down here, you can see the little openings of the submandibular ducts there as well. Let's see. What do I want to do on here? You can see at the top here, you have the hard palate that is on the top there. And that hard palate is basically formed by the palatine bone and that palatine process of the maxilla with a kind of this midline ridge up there. You have the soft palatine that comes down here. Mostly those are folds of skeletal muscle, if you're wondering. And this guy is going to be what ends up closing off the nasopharynx during swallowing. This area, that's actually these guys that come up and you see those two arches. So the you can see that comes down as the uvula, right? And let's see if there's anything else that I want to do in here. Sorry. You can see the tonsils back here in this area. The palatine reef is this area that's connected between the two. Basically helps to, that's that midline ridge in the hard palatine area. And I think we're good with that for now. Now, if we're looking at the side here, you can see, and so, let's see if we're going to talk. Let me look forward really quick. Yeah, I'll talk about the salivary glands here in a minute. And so you can kind of see this back area. So when we're talking about, you can see the tongue here. And so this is the oral cavity that is kind of this mid-sagittal cut that you're getting. So you can see the oral cavity here. You can see the uvula going down here. Of course, we're going to talk about all the different salivary glands. But if you notice, you have this area that's back here, which is the orthopharynx area. And that's the area that's going to be cut off and separated so that swallowing can happen, right? And then you can see down here in this region is the laryngeal pharynx, which we're going to talk a little bit more about when we get to breathing. And so, And then, of course, you have the trachea here and the esophagus here. And basically, we're going to have the epiglottis that will cut this off here so that everything can go down there versus going down to your lungs. And so this is kind of more of that kind of general anatomy of the mouth on here. And so when we're talking about the mouth, right, a big part of the mouth is the tongue. It occupies the floor of the mouth. And basically, it's composed of bundles of skeletal muscles. And it has basically 3 functions. It's important in gripping, repositioning, and mixing of food. And this happens during chewing, right? It's important in the formation of the bolus. And so the bolus is this mixture of food and saliva. And it's pivotal in the initiation of swallowing, speech, and taste. Now, the muscles that are important in changing the shape of the tongue are the intrinsic muscles. And the ones that help to alter the tongue's position are the extrinsic muscles. The lingual frenulums are attached to the floor of the mouth, and that helps to kind of stabilize them. So let's look at the papillae that are visible on the tongue. And there's different ones, right? So the first one that we're going to check out here are these ones here. We have the filiform papilla. So these guys right here are important. And what these all are these peg-like projections. And they're kind of this underlying mucosa that are in the mouth, right? So this gives the tongue roughness. Let me just put roughness. Right? So that roughness is going to provide friction. This is not a taste bud. And this can give the tongue a whitish appearance. The next one that you can look at here is the fungiform papilla. This guy is mushroom shaped. It's scattered all through the tongue. This is what gives the tongue the reddish appearance. The valate papilla, also known as the circumvallate papilla. These guys, they're about 8 to 12 V-shaped rows. And they're on the back. And the folate. You can see those right there. Papilla are located on the lateral aspect of the posterior tongue, right? You can see them over here on the side. Now, if you look on here, guys, you can see this guy right there, the terminal sulcus. That's right back here, right? So this guy actually marks the division between what's considered the body and the root. And so the body is the portion of the tongue that's in the oral cavity. And the root is the part that resides in the oropharynx. Now this guy does not contain any of this papillae. It seems rough, but that's because of this tonsil, the lingual tonsils back there. But it doesn't have any of those. So those are the different parts of the tongue, or at least those peg-like projections. Now, within our mouth, we're going to have salivary glands that essentially produce saliva. And the function of saliva are many. For instance, it helps to cleanse the mouth. It helps to dissolve food chemicals for taste. It moistens food. So it basically makes a bolus. And it begins the breakdown, specifically of starch. And the enzyme that does that is amylase. So most of the saliva is produced by the major extrinsic salivary glands located outside the oral cavity. Minor salivary glands are scattered throughout the oral cavity, augment slightly. So let's look at those salivary glands. So just to give you an idea, saliva is mostly, let me see. Saliva is mostly water. Okay? But it does contain electrolytes, amylase, specifically salivary amylase, right? What's known as lingual lipase. It's going to have mucins, lysozyme, and antibodies. And of course, there's always some waste in there, metabolic waste, right? So when we're talking about salivary glands producing saliva, we have a couple. We have basically a few that we're going to look at here. You can see the very first one is this parotid gland. You can see it's up here. Here's the masseter muscle. So it's back here towards the back of the ear. Submandibular gland, think about the mandible towards the bottom. That's going to give you an idea here. And sublingual, which is around the tongue. So those are the three main types of salivary gland, and that are going to be basically producing saliva. Now, when we talk about the production of saliva, it's basically salivary glands are composed of two main types of secretory cells. There's serous and mucus. And so when we're talking about what saliva is made up of, the serous actually produces the watery secretions, the enzymes, the ions, and a bit of mucin. The mucus cells produce mucus, right? So when we're talking about those parotid and submandibular glands, contain mostly serous types of cells. So underneath, towards the back and towards the mandible, basically they're going to be producing all the watery secretions, enzymes, and all that. And the sublingual, which is closer to the front, right, it's going to catch most of the bad stuff, is the mucus, right? So think there, it's towards the back, or It's closer to the front, right? So it gets the bad stuff. These guys, closer to the back, digestion. That can help you remember the two. So just a reminder, saliva is mostly made up of water, so it's hypoosmotic. It's slightly acidic. It has electrolytes in it. It has different types of enzymes, salivary amylase and lingual lipase. It produces proteins like mucin, lysozyme, and IgA. IgA is another nice way of just saying antibodies in general, right? It has some waste. And just remember, there's all kinds of stuff in here that we've talked about previously to help against microorganisms, because that's an opening inside our body. So they're going to have to put some kind of defense mechanisms to make sure that we're not letting anything harmful to us in through that way. So how much do we, when we're talking about salivation in general, how much do we produce? Well, about 1,500 milliliters per day, or 1.5 liters, right? Basically, you have minor glands that will continuously work to keep the mouth moist. And the major salivary glands that we talked about are activated by parasympathetic nervous system. And those are activated when basically you have ingested food. Right? When you have that ingested food, they're going to activate either chemo or mechanoreceptor. And they, once that happens, they like to talk to what's known as a salivatory nuclei in the brainstem. And this activates the parasympathetic, which means increased digestion. And so you can get xerostomia if you have a strong sympathetic stimulation, which means if you're going to have a strong sympathetic, that's going to inhibit digestion. So that essentially will inhibit salivation and can produce dry mouth. Now, the problem is if you're not making saliva, you're also not making those molecules or releasing those molecules to help to basically clean out your mouth and help to promote getting rid of bacteria. So it can produce or promote tooth decay. And what's funny is we know sight, smell of food, or epsite GAA can act as a stimuli as well. So teeth, they perform mastication, and that's basically chewing in that sense of performing mechanical digestion. Dentation and the dental formula. So basically, you have primary dentation, which is talking about having baby teeth. So you have 20 deciduous teeth, which are the milk or baby teeth, and basically they are there within 6 to 24 months of age. Then you have secondary dentation. Basically on here, you have 32 permanent teeth, and they come out and occur between the age of 6 to 12 years. All but your third molars, which are wisdom teeth. Some people don't even have wisdom teeth. For instance, my son doesn't have a set of them. They're normally in by the end of adolescence, between 17 to 25, but sometimes the wisdom teeth can come out later in your 20s, later 20s, early 30s, but most of the time it's between the age of 17 to 25 years of age. And so let's look at the types of teeth we have and point them out. And so we have incisors. These are basically those kind of chisel shaped that will help to cut food. They have one root towards the front. The canines are towards the side, and they're fang-like, and they tear, they have pierce. They have one root. Premolars, which are also called bicuspids. These are broad crowns. They're rounded cusp used to grind or crush. They normally have one root, except the first upper premolars. And then the molars are these broad crowns. They're rounded. They're best grinding, right? And your first two upper molars have 3 roots. The first 2 lower molars only have 2. And the third molars vary. You don't need to know how many roots that they generally have. I just find it interesting that they have all these different roots. So you can see, and basically they're talking about with the deciduous or the baby teeth when they come out. So basically they don't all come out at one time, and we all know this because you've been a child or you have children yourself. The central incisors are the first to come in. There's normally about 6 to 8 months. And then you'll have the lateral, which are the second at 8 to 10 months. And then basically you'll have your first molars, which are back here, which are about 10 to 15 months. Then you'll have your canines come in, also known as eye tooth, between 16 and 20 more. And your secondary molars are your last around 2 years. And then again, these all fall out and get replaced by your permanent teeth. So as you can see with these guys, it's roughly about the same. These guys come in first, right? So your central incisors come in around the year 7. And then you get your lateral that will come in. You'll get your first molars coming in around 6 and 7 as well in the back. You'll have your next coming in are your canines around 11. Your premolars here around the second time. And then all of these kind of do the second premolars, your second molars, and then these guys at the end are last if they come in at all, or if you even have them, which are your wisdom teeth. Again, don't need to know what year they come in, right? What's important really here is what their functions are, and can you identify where they're located in your mouth. And so this kind of gives you an overall idea of what the tooth structure actually looks at. And once we're done with the teeth, we're going to take a break. And so each tooth has 2 major regions. You have the crown region, right? And then you have the root region here. And so the crown is the exposed part that's above the gums, right? It's covered by enamel, the hardest substance in the body, right? And so this guy is very much has heavy mineralized with both calcium salts and what are known as hydroxyapatite crystals. And we talked about this when we were talking about bone, right? So enamel is not replaced. Enamel producing cells actually degenerate when tooth erupts. So no healing if the tooth decays or cracks. You need artificially replaced by filling, right? We know this. And so you can see the enamel here, and then on this inside, basically you'll notice the dentin here. The dentin is a bone-like material, right? It's maintained by, you'll see that you notice that you'll have the pulp cavity that's right here, and you have what's known as odontoid blast. Think blast cells and orthodontists, odontoid blast will maintain that dentin or bone-like material underneath the enamel. Now you have the pulp cavity. This is where the blood vessels and the nerves are. And then basically, within that is pulp, right? And this is just connective tissue, blood vessels, and nerves. And then you can see as it goes down, you have the root canal there that goes down to what's known as the apical foramen, right? This is on the proximal end. And this is the entry, right, of basically blood vessels. I'm going to put BV for blood vessels and nerves. Now you can see from all this is part of what's known as the root. And so this is a portion that's actually embedded into the jawbone. And these guys are connected to each other by the neck. So on here, if you remember, the periodontal ligament is here. And if you remember, what kind of fibrous joint was this? Hopefully you remember the gamphosis joint, right? And so this periodontal ligament that's there actually anchors the tooth in that socket, right? And basically this what connects the periodontal ligament here to the actual root is the cement, and the cement is just calcified connective tissue. And so that kind of gives you an idea of the teeth and what the teeth are important. So at this point, I'm going to take a bit of a break. So we're going to go, we've just finished the mouth, talking about the tongue and the teeth, and now we're going to go into the pharynx and the esophagus. So after we swallow, basically, it's going to go from the mouth into the oropharynx, into the laryngeal pharynx. And then once it goes through that area, it's going to go into the esophagus next. Now, the esophagus is basically a flat muscular tube. And it basically goes from that laryngeal pharynx to the stomach. This guy is basically collapsed when it's not involved in food propulsion. It'll go through the diaphragm at an area that's called the esophageal hiatus. And basically, it's going to connect to the stomach at the cardial orifice. Let me go up here. Now, when we look at this, the esophagus does have all 4 alimentary canal layers. And basically, you have within the esophagus glands called esophageal glands. They're located in the submucosa, and they help to secrete mucus to aid in the movement of the bolus. And again, if you remember, there is the area that's called the muscularis externa, and this has skeletal muscles, basically that help to mix, and then smooth muscles in there as well. And so that helps with basically the movement or the propulsion with the peristalsis and the mixing that happens less likely in the sedimentation. Yeah. Great. Move forward. Sorry about that. So here, let's look at the layers of the esophagus here. You can see the lumen in the middle, and then on that interior is the mucosa that is in that area. And basically, just to let you know, there's stratified squamous epithelial, and it does change once it hits the stomach to simple columnar. Not truly important to know, right? Then we're going to have the submucosa, and this is where the basically mucus gets secreted in this area, and that's going to help to basically move the bolus within the esophagus. Then, of course, you have the muscularis externa, and this is what helps with the movement of the bolus throughout. And here, instead of the serosa, you have that inventita. And basically, this is just more robust. And so those kind of give you the different layers of the esophagus. So, the pharynx and the esophagus are conduits to pass food from the mouth to the stomach, right? And basically, the major function of both organs is propulsion, and that's essentially to get the food that becomes a bolus in the mouth through from the mouth to the stomach. And that starts with deglutition, and that's the process of swallowing. This involves the coordination of about 22 muscle groups and 2 phases, the buccal phase and the pharyngeal esophageal phase. And so the buccal phase is basically the voluntary contraction of the tongue. And basically, the pharyngeal, esophageal phase is in the voluntary phase. This mostly involves the vagus nerve. And it's basically controlled by swallowing centers. And those are located both in the medulla. Something's wrong with your medulla oblongata. And the lower pons. So let's talk about that process of swallowing.
The oropharyngeal and esophageal stages. At the start of a swallow, a food bolus is voluntarily pressed by the tongue up against the roof of the mouth and backwards towards the pharynx. In response to activation of pharyngeal pressure receptors, the swallowing center in the medulla initiates reflexes that prevent food entry into respiratory passageways. The uvula contracts, which blocks the nasal passages from the pharynx. The laryngeal muscles contract, closing the glottis at the top of the trachea by tightly aligning the vocal folds. The epiglottis swings down upon the closed glottis. With all airways blocked off, respiration is temporarily inhibited. As the upper esophageal sphincter relaxes, pharyngeal contractions drive the bolus into the esophagus. The oropharyngeal stage is done, and breathing resumes. During the esophageal stage, a primary wave of peristalsis initiated by the swallowing center pushes the bolus through the esophagus. As the bolus travels through the esophagus, the lower esophageal sphincter relaxes, allowing the food to enter the stomach. If the bolus is sticky and adheres to the esophagus, secondary peristaltic waves, triggered by the intrinsic plexus at the point of distension, completely clear the esophageal lumen to finish the swallow.
And so if you look at this, guys, this area, let me get a different color here. This area that's right here, where the basically the esophagus joins the stomach, that's what's known as the cardinal orifice. And in there, that closes it off, right? That keeps this opening or this orifice closed is the gastroesophageal sphincter. It's also known as the cardiac sphincter. So this surrounds the sphincter, surrounds the cardinal orifice, and it keeps the orifice closed when food's not being swallowed. Mucus cells on both sides of that sphincter help protect the esophagus from acid reflux. Okay? And so, if you look at here, you have two phases, right? You have the buccal phase, which once you allow food here, right, the first thing you're going to do is it's going to have this upper esophageal sphincter is contracted or closed. And you can see that right in that area that I've pointed to. The tongue will press against the hard palate and force the food bolus into the orthopharynx. And so the orthopharynx area is that back area right there. So that's going to be the buccal phase that happens first. And the pharyngeal esophageal phase begins once the tongue blocks the mouth. You can see that there. The soft palate and the uvula will rise, closing off the nasopharynx, which is that area at the top, right? So that it can't go up into your sinus cavities. The larynx rises, so the epiglottis blocks the trachea, and so you can see that in that area right here. And the upper esophageal sphincter relaxes, which was originally right in this area, right? It allows the food to actually enter the esophagus, and you can see it'll go down there. And as this phase continues, basically the pharynx will contract, forcing food into the esophagus as it goes down. And that upper esophageal sphincter will contract again once the food starts its way down the esophagus. From here, peristalsis will move the food through the esophagus to the stomach. And you can see at this point that the esophageal sphincter is closed. That's also known as the cardiac sphincter, right? And basically the food or the circular muscles will contract, pushing the food through that sphincter, and it'll open. And that sphincter closes around the cardial orifice, right? So that orifice opens. And then after the food enters the stomach, the sphincter will close, preventing regurgitation or all those juices coming into your esophagus. And so I thought this was kind of cool if you wanted to see what it looked like with someone actually swallowing. So here there, you can see the food moving around from the stomach. And see all of this. And you can see some of that going from the buccal phase, the very beginning, and then once it starts moving down, going into that pharyngeal. What's there? You can see everything in there.
Recording.
So that kind of gives you an idea of what it looked like. Now the next part that we're going to talk about is once it gets through that orifice into the stomach, what happens? So the stomach is the temporary storage tank, and this is where much of the chemical breakdown of protein starts, or protein digestions. Carbohydrate digestion started in the mouth with the amylase that you have. And so basically what's happening here is we're going from that bolus, and the stomach is producing chyme. An empty stomach is somewhere about 50 milliliter volume, but it can expand up to 4 liters when you're basically eating. And as you can see these little lines in here, and they are called root. And basically these are the stomach folds, right? They help to increase that surface area. And so let's look at it and get a general area. So you can see this is the esophagus. So that opening right here is the cardial orifice. So you can see this area towards the top is called the cardia. And so this area is what actually surrounds the cardial orifice. Now, if you look at this dome part at the top, this is going to be right beneath the diaphragm. Diaphragm is up here, right? That's going to be what's known as the fundus. Now, the midpoint of the stomach is the body. And then you can see at the end is the pyloric area. This is wider and more superior part of the pyloric region. So you can see the pyloric atrium, the pyloric canal here, and then the pyloric sphincter over here. So basically it starts with the superior region, which is the antrium here, and then it'll go through, and basically the canal gets even kind of more narrow, and then it terminates in the pylorus, specifically this region that's called the pyloric sphincter at the pylorus. And so this is what closes, right? And this pyloric valve or sphincter controls the emptying of the stomach into the small intestines. That area is specifically called the duodenum or duodenum. Now the layers are similar here, and you can see that in the stomach you have the circular layer, the oblique, and the longitudinal layer of all these different basic muscles in here, right? And so as we're going through here, you can even see they're talking about the greater curvature area that's down here in this, right? This is that kind of convex lateral surface of the stomach. The lesser curvature is on the other side. As you can see, that curve is a little bit kind of smaller, and this runs from basically the lesser, well, this is the concave medial surface of the stomach. The mesenteries extend from this curvature. So this is, you're going to get the mesenteries extending from this side, right? And tethering the stomach to other digestive organs. I have it on here. At this point, we're going to, you can go the omentum. There's the lesser and greater omentum. So the omentum, or specifically the lesser omentum, is going to basically run from the lesser curvature to the liver. And the greater omentum is going to drape from the greater curvature, right? And basically this is going to drape over the intestines, the spleen, and the transverse colon. So that's basically the anatomy of the stomach.
What exactly happens in our stomachs when we eat?
Even before we have taken the first bite of a meal, the brain sends impulses to the stomach. These impulses start the production of secretions in the stomach, and the upper part of the stomach, which acts as a reservoir, expands. The food passes into the stomach through the cardia at the stomach entrance. This is a muscle that acts like a valve, closing the top of the stomach. The top part of the stomach, the fundus, is where food and the air that we swallow with every bite is collected. This is where the stomach volume starts to adapt. The fundus is an extremely adaptable structure. The more food goes into the stomach, the more it actively expands. In the middle part of the stomach, called the body, gastric juice is produced and mixed with the mass of chewed food. The main component of gastric juice is gastric acid. This eliminates bacteria in the food and also helps to prepare the food for the following stages of digestion. The muscular contractions of the stomach wall churn the food mass and mix it with gastric juice. 3 contractions per minute move the food mass back and forth. After a certain time, the contents of the stomach are adequately broken down and mixed, and are passed in small quantities into the intestine via the pylorus. As the stomach empties, it gradually returns to its previous size.
Bear.
So that kind of gives you an idea of how the stomach works and what we're going to be talking about for this time. So the control and the blood supply of the stomach. The autonomic nervous system supplies the stomach. So you have sympathetic fibers that originate from the splenic nerve in the thorax that are actually relayed through the celiac plexus to help with supplying the control of the stomach. Parasympathetic fibers are supplied by the vagus nerve if we're going to start or stop digestion, right? And the blood supply that comes from the stomach essentially is the celiac trunk. That's the gastric and splenic branches. And then, blood that leaves from there goes from the veins of the hepatic portal system, because again, we're going to want to clean that blood before we take it and put it back into circulation. So that's basically how we control the stomach, through either generally through the autonomic system and how the blood gets to the stomach and back out. Now, when we're looking at the stomach microscopic anatomy, right, the stomach wall actually contains 4 tunics or 4 layers. And you can see here, it's the same as what we were talking about earlier when we were looking at the general anatomy of the digestive system, right? The mucosa that has both the epithelium, the lamina propria, and the muscularis mucosa. Then you have the submucosa area that contains the submucosal plexus, right? And then you can see the muscular externa, which contains the myenteric plexus, and you can see all the different layers there in the serosa. I'm not going to go into a bunch of detail here because I've already kind of talked about how this works. The only thing that I'm going to go into detail are just a few of these so that we can tell what's going on. For instance, in the stomach, or the muscularis externa, we have the 3 different layers, right? And so this when we're looking at, we have the circular and the longitudinal smooth muscle layers, but we also have an extra 3rd layer there, right? And so when we're looking at the oblique layer there, right? This is the inner layer there. This allows the stomach to not only churn, mix, and move chyme, but it also allows it to pummel it. So this increases physical breakdown. So that's how this is. The other layers just have these 2 guys, right? Where the stomach has this oblique inner layer that helps. Now, these guys are also a little different when we're looking at the mucosa layer, right? Because this actually makes a 2-layer coat of alkaline mucus. And it makes sense because we are producing in here so much stomach acid to break things down that we want to make sure to protect the lining of the stomach. Okay. And so now that we're looking at this, let's look at these. This lining produces all these gastric pits here, right? You can see these openings here, and then this is one of them that we're going to look at. So basically, this is the mucosa layer. And basically, that layer, like I said, is a little modified. It consists of all these basically cells, or this layer that's composed of entirely of these mucus cells. And so you can see all of these mucus cells that are there. Again, if you remember, mucus cells help to produce basically a thin, acidic mucus. Now, they don't really know what its known function is at this point. Now, if you look, these little purple one, or these little turquoise ones right there, are parietal cells. And so they secrete two things. They secrete hydrochloric acid. So the hydrochloric acid is somewhere between pH 1.5 and 3.5. And this has a few things that it does. So let me go over here and write this. And then I'll go back. So this helps to denature proteins. It helps to activate a protein called pepsin, which is going to help to break down proteins. It's going to break down plant cell walls. And it's there to help kill bacteria. Not all, but a lot of them, right? So that's what hydrochloric acid does. Now, to let you know, hydrochloric acid is not the only thing that this parietal cell secretes. It also secretes intrinsic factor. And this is a glycoprotein, and it's required to help with vitamin B12 absorption. And this happens in the small intestines. So we've covered the mucous neck cells, we've covered the parietal cells. Now let's talk about the chief cells. Now the chief cells secrete two things. The first thing they secrete is pepsinogen. And so this is an inactive enzyme that basically gets activated by pepsin. Or it gets activated to, sorry. That basically, oof, why does it do that? It's an activated enzyme that gets changed into pepsin due to HCl. So the chief cells secrete pepsinogen. The parietal cells secrete hydrochloric acid. And once pepsinogen comes in contact with hydrochloric acid, it becomes activated into pepsin. It also secretes some lipases. And this digests lipids, about 15% of them. Now you do have the enteroendocrine cells down here at the very bottom, right? And so these guys do a couple things. They secrete chemical messengers. Into the lamina propria, right? So they act as these things called paracrine cells. If you remember in part 1, we talked about these. These act on nearby cells, right? Versus autocrine that acts on the same. Now, they're responsible for secreting serotonin and histamine. So these are a couple of basically hormones that they secrete. In addition to, they also secrete somatostatin and gastrin. And we're going to talk all more about what all these guys do later. So these essentially are, these gastric pits open into this area, and these are responsible for producing out of here, these mixtures of cells produce the gastric juices that get released into the stomach. And it's a combination of all these guys working together to produce this, this juice to help with breaking down of helping to break down the food within the stomach. Now, this endocrine cell that produces basically this gastrin and the somatostatin, these guys are important because essentially these help to produce a thick layer of bicarbonate rich mucus. Can you see why that might be important? If we're producing all this stuff that's going to basically make the stomach really acidic, which will cause items within the stomach to just go through hydrolysis. If we're producing these enzymes that will start tearing away proteins and fats and all this, we want to make sure that we're not actually digesting the cells themselves that are in the stomach. And so here, basically, we want to make sure that everything is working the way that it needs to. So these are going to work together to basically kind of protect or to digest. So the mucosal layer that we have that gets produced protects the stomach and is created by three factors, basically. Again, a bicarbonate layer, what else also helps us are there are tight junctions in the stomach. It's between the epithelial tissue, so essentially it's going to prevent the juice from seeping underneath the tissue. And basically, mitosis is fast. The surface of this is replacing every 3 to 6 days to help make sure that the damaged epithelial cells are removed quickly. So if anything breaches this mucosal barrier, it can lead to gastritis. And this is an inflammation caused by anything that actually breaches the stomach's mucosal barrier. So things like peptic or gastric ulcers are considered this because they can cause erosions on the stomach wall. And if these erosions actually perforate the wall, it can lead to peritonitis and hemorrhaging, right? And most of these ulcers are caused by Helicobacter pylori, which is a bacteria. And the guy is very interesting that actually hypothesized this. People thought he was full of just nonsense forever. And so what he actually did is he had this idea that he felt like this bacteria caused ulcers. So he infected himself with this bacteria, H. pylori. Then he got ulcers, and then he gave himself an antibiotic to kill this, and his ulcers went away. And then he released and published that data. So it's kind of interesting. They can also be caused by non-steroidal anti-inflammatory drugs, things such as aspirin. And so this kind of shows you the H. pylori bacteria and the mucosal layer of the stomach, and then essentially what an ulcer can look like. And so the digestive process is carried out by the stomach in basically these 6 steps. Basically, it carries out broken down food. It serves as a holding area. It delivers chyme to the small intestines. It denatures proteins, i.e. breaks them down, right, by hydrochloric acid. Pepsin actually carries out protein digestion. Now, casein, which is a milk protein, is a little bit different. It's actually broken down by renin in infants. And so that's why when infants like spit up and it has that curdy kind of substance in milk, that's why it's a little bit different. Now, basically, lipid-soluble alcohol and aspirins are absorbed into the blood through the stomach. And the only stomach function absolutely essential to life is the secretion of intrinsic factor. And if you remember, intrinsic factor is for the absorption of B12, vitamin B12. And if you remember, why is vitamin B12 important? Because it's needed for red blood cell maturation, right? And it can lead to pernicious anemia if you lack this intrinsic factor. So these are all the different digestive processes that are actually carried out by the stomach. So how do we regulate gastric secretion? Well, gastric mucosa secretes about 3 liters of gastric juice per day, and it's regulated by the following. You have neural mechanisms. So basically, the vagus nerve stimulates the increase in secretion, and the sympathetic stimulates the decrease in secretion. You also have, besides neural mechanisms for gastric secretion, you also have hormonal. So gastrin helps to stimulate enzyme release and HCl secretion. So the parietal cells that secrete HCl, gastrin released by the endocrine cells help to increase that. Now, gastrin antagonists are secreted by the small intestines. And this makes sense because if it's starting to get into the small intestines, then you can slow down on the production of enzymes and HCl secreted in the stomach, right? Now, gastrin secretion are broken down into 3 phases. You have the cephalic phase, you have the gastric phase, and the intestinal phase. And so the gastric or cephalic gastric phase for gastric secretion is a conditioned reflex. So think things like aroma, taste, sight, thought. All those first things that happen when you're thinking about eating, right? That's going to be the cephalic or the reflex phase. The gastric phase basically lasts about 3 to 4 hours. This provides about 1/3 of the gastric juices released, right? And so how does this gastric phase get stimulated? Well, basically you get swallowing, distension or swallowing activates these stretch receptors. And that's going to initiate both the brain, right, and short reflexes, which is your gut reflex. So that's one thing that's going to stimulate it. You can also get chemical stimuli. So things such as partially digested protein, caffeine, low acidity. will activate those endoendocrine cells to secrete gastrin. And basically, once those chemicals, let's say the endocrine cells release gastrin, and basically that release of gastrin then is going to trigger HCl release from the parietal cells and activate enzyme secretion. So that's stimulation of the gastric phase, right? Now, once we have basically those endoendocrine cells release not just gastrin, but they also release histamine. And histamine's there to help basically buffer the action of ingested proteins, because those are going to cause a pH to rise, which activates more gastrin. And so this is going to kind of help to buffer some of that action. Now, how can we inhibit the gastric phase? So basically, low pH does. So pH is going to lower between things like between meals, it can happen during eating due to negative feedback. So basically, this may not sound like it makes sense, but think about it. The more protein that you have, the more HCl that's going to be secreted, because HCl is going to, or hydrochloric acid is going to activate pepsinogen into pepsin, which then can break down proteins. Well, that's all fine and good, but it's going to cause a decline in your pH, which will inhibit gastrin secretion over time. So those are two things. Basically, an inhibition of gastric phase is mostly based on low pH, right? Now, the intestinal phase it begins with a brief stimulatory component followed by inhibition. So basically what's going to happen is your food, your partially digested food, is going to enter the intestines, causing a release of enteric or intestinal gastrin. And so this is going to encourage the gastrin glands of the stomach to continue secreting activity. And so basically, let me write this down. So partially digested food, right, goes into small intestines. I'm going to basically, that's small intestine right there. And so this is cause a release of the intestinal otherwise known as enteric gastrin. And so basically you have partially digested food, maybe not all the way, it gets into the small intestine, so it's going to tell the stomach, hey, release it, release more gastrin, gastric juices into the stomach so we can continue secreting the activity and breaking things down. Now, when do we stop this? When in the intestinal phase, what inhibits this? There's four main factors in the duodenum, that area that it hits right after it gets out of the stomach, that causes inhibition of gastric secretions. The first is the distension of the duodenum. So this is the basically the stretching. And this is due to why is this so? I'm really sorry. Oh no. Of duodenum, and this is due to the chyme actually entering it. Another thing is the presence of acidic chyme, fatty chyme, or hypertonic chyme. So these inhibitory effects actually protect the intestines from being overwhelmed by too much chyme or too much acidity that it can't actually take, right? The inhibition is achieved in a couple different ways. And that's what we're going to talk about next on our next slide. So it's inhibited by the enterogastric reflex and the enterogastrons. So the enterogastric reflex is in the duodenum. It actually inhibits acid secretion in the stomach by interacting with the enteric nervous system, which is the short reflex, and the sympathetic nervous system and vagus nerve, which is the long reflex. So basically gut brain, brain, right? And it also works with the duodenal enteroendocrine cells, which release two important hormones that inhibit gastric secretion, and that's secretin and CCK, or cholecytokinin. So let's see what both of these guys do. Just to let you know, basically how all this intestinal phase works is that the stimulatory effects is very brief. So when I'm talking about stimulation happening here, it's very, very short time. It's towards the beginning as stuff is starting to enter in the duodenum, but it's overridden by the inhibitory stimulation as the intestine starts filling, right? And so here's the CCK, and you can see hormones in the paracrine that act in, and we talk a lot about different ones, but this is used to help basically with the inhibition of some of the gastrin released during the intestinal phase. And so the site of production is in the duodenum, in the mucosa of the duodenum. It's basically fatty chyme and partially digested proteins that help with the stimulus for production. And basically these are all the different areas for which it'll target. And basically it helps, the main part of this that we're going, it inhibits stomach secretory activity. So it basically helps to reduce the gastric juices that are being produced in the stomach once we get into the intestinal phase. And the other one is the secretin. Again, you see it made in the mucosa of the duodenum, and its stimulation is partially digested proteins and fats, again, basically acidic chyme, and its target is the stomach, pancreas, and liver. And in here, as you can see again, it inhibits the gastric gland secretion and gastric motility. Okay? And so these are the two things. Of course, there's other things in here that they're talking about, but those are the two main that we're focusing on in the looking at the inhibition of the intestinal phase. So let's look at the neurohormonal mechanisms that regulate or stimulate or inhibit gastric juices. So this is just a review of what we just talked about. So the cephalic phase, basically when you're looking at this site and thought of food, goes to the cerebral cortex, through the hypothalamus and the medulla oblongata, through the vagus nerve, into the tummy, and basically this is going to start with gastric juice production. Add to that also taste and smell receptors communicate, and all of this goes down and starts producing basically gastric juices. Now, and once that happens, you can see on here the gastric phase, your stomach actually starts to stretch, that distension. I think I said swallow earlier, but I meant to say stretch. It activates those stretch receptors, and basically your long reflex, which is through the medulla and vagus nerve again, so we're looking at the brain, will go in there and actually activate gastric juice production. And then if you look, the short reflex, which is the gut brain, will come in there and activate that as well. The second thing that's important on there are food chemicals like peptides and caffeine. They're going to help raise the pH, which will activate chemoreceptors, activate the G cells, and the G cells are those enteric endocrine cells that will release gastrin to the blood, and that will activate gastric acids in the stomach, right? And then once you get through the tummy area and you're going into the duodenum, we'll get into the intestinal phase. And basically here, once you get a small amount of partially digested food into that area of the duodenum, or distension of the duodenum, a bit of stretching, when the stomach begins to empty into there, you'll get a brief effect where it will do the intestinal enteric release of gastrin to the blood. This is brief though, right? Immediately after that, you get into the basically the inhibitory effects. And basically with here, once you get a certain amount of stretching of the duodenum, a presence of fatty, acidic, or hypertomic chyme, then you're going to get a inhibitory effect. So you're going to get secretin in that CKK, cholecystokinin, or CKK. that gets released, and that's going to basically, these are entogastrons that will reduce the secretion, or the intergastric reflex, which both includes the both short and long reflexes, both gut brain and regular brain, to stop. Now, when we're talking about the inhibitory of the gastric phases, basically, if it becomes too low, or the pH is too low, then essentially what you can see is the G cells will basically reduce or decrease the amount of gastrin released to the blood, and so that will reduce the amount of gastric juices secreted. Also, emotional stress can activate the sympathetic nervous system and cause you to override the parasympathetic and reduce gastric juices. And to go with emotional stress, loss of appetite and depression can also cause a decrease in parasympathetic activity and inhibit. So how is HCl secreted by the parietal cells to begin with? Because this is a big part about how things are secreted. Give me 1 second. So, you can see we're taking a bit out here of HCl and what happens. So here's the blood capillaries, the interstitial space right here, and the actual gastric gland. And so if you look at the first, we're going to have both bicarbonate and HCl are generated from the dissociation of carbonic acid produced from CO2 and H2O. And so if you look here, you have water that's available here, and you have carbon dioxide coming in. So you have all of this carbon dioxide, basically all of these different things happening or coming into the blood, which comes over into this interstitial fluid. And so the CO2 is going to combine with the water to make carbonic acid, which is right here, right? And basically, the carbonic acid is going to break down into basically an H and the bicarbonate. From here, this hydrogen or proton is going to be pumped using the hydrogen potassium ATP pump into the lumen of the stomach. Of course, the K, or the potassium, returns to the lumen through the membrane channel. So you can see on here, it's going here and then it's coming back. Right. The main point of this is to try to get the H on the other side. Now, this guy, the next part that we're looking at is within the interstitial fluid, you also have calcium. And so on here, we're getting calcium to come in and bicarbonate to leave, because that's how this pump works. So you're getting basically bicarbonate going out to help buffer the blood. And then from that, we're able to get chlorine in. And so this is going to diffuse into here and basically make HCl. So when we're talking about how we're getting HCl made by these cells, right, what you can do is the H is coming from the breakdown of carbonic acid. That's coming from water and CO2, right? And the Cl is coming from basically the blood and the interstitial fluid, and it's an exchange between the bicarbonate and the Cl. Right? So that's how it's working, and that little cell is doing all that work just to get hydrochloric acid. So when we're talking about gastric filling, or how we regulate gastric filling, it's a response of the stomach to filling. So there's basically stretch receptors there, right? And so in your stomach, you can keep bringing food in because you're able to accommodate the stretching, right? And there are two factors that cause pressure to remain constant until we get 1.5 liters of food is ingested. So you have receptive relaxation and you have gastric accommodation. And so receptive relaxation is the idea that the stomach, you can relax the stomach due to swallowing. So as you swallow, it's going to relax to cause the stomach to be able to fill. And basically, this is a coordinated effort using the swallowing centers in the brainstem. So the gastric accommodation is this idea that it stretches to decrease tension and then it relaxes. It's kind of the idea that if you're stretching for the splits, you stretch for a while and then you relax, and then you can stretch longer the next time, right? So this is what's known as a stress relaxation response. And this is what allows the hollow organs to stretch without increasing tension or contractions. So that's how you're able to get more food while you feel. How do we allow the motility and the emptying of the food once it gets in there? Well, you have gastric contractile activity. So you have this peristaltic wave that moves towards the pylorus at a rate of 3 per minute. You have what's called a basic electrical rhythm, or BER, is set by what's known as these things called enteric pacemaker cells. Think of the same idea as the heart, that the heart could basically contract on its own because it had its own pacemaker cells. So the stomach does as well. They're linked by gap junctions, so the entire muscular contracts together. Distension and gastrin increase the force of contraction. So the more it stretches and the more gastrin that gets released, the more it increases the force of the contraction to try to empty it. Contractions are most vigorous and powerful close to the pylorus, or that region where it's the pyloric sphincter, where it's emptying, right? And 30 milliliters of chyme is produced. And so when it's delivered to the duodenum, it's delivered in about 3 milliliter spurts. And then the rest of it, the 27 other mil, is forced back. And this little valve, this pyloric valve that you're going from the stomach to the duodenum, only liquids and very small particles are allowed to pass through it. And so when you're looking at this, the peristaltic waves in the stomach, how we move, you can see it contracts here, and so you have propulsion. That's this peristaltic waves move from the fundus up here down to the pylorus, which is here, right? And so you can see that it's starting to move down. And basically, then you have grinding. And so that's the most vigorous peristalsis. And mixing action occurs closest to here, the pylorus, right? The pyloric end of the stomach acts as a pump that delivers small amounts of chyme into the duodenum. Remember, there's 30 mils, but only 3 mils go at a time. And so that bunch of that doesn't get to come out gets forced back. So it's called retropulsion. And the peristaltic wave closes the pyloric valve here, forcing the contents of the pylorus back into the stomach. So that's going to essentially help to mix that stuff better as well. So as we're emptying the stomach, how does the duodenum prevent from overfilling? And basically, they prevent from overfilling by controlling chyme entry. And so if you remember what we were just talking about, you have in here duodenal receptors in that area, in the duodenum, right? That respond to stretch and chemical signals. You also have both the reflexes, remember? The enterogastric reflex and introgastrons. Secretin in the CKK that's there that inhibit basically gastrin or gastric secretions, right? And the filling. So the stomach will empty in about 4 hours, but increase in fatty chyme entering duodenum can increase time to about 6 hours or more. Right? And then you have the carbohydrate-rich chyme moves quickly through the duodenum. And we are so close to the end, guys. Just hold on just a few more. So on this emptying factor, right, the neural and hormonal factors that inhibit gastric emptying, again, any kind of presence of chyme that's fatty, hypertonic, or acidic, right, that's going to come, and that's going to basically going to be picked up by the chemoreceptors and the stretch receptors, and also the duodenal enteroendocrine cells. They're going to secrete the CKK or the secretin, and that's going to act in reduced contractile force on the stomach and reduce the rate of stomach emptying, aka the amount of gastrin produced as well. On here, those chemoreceptors and stretch receptors will activate or trigger the enterogastric reflex. The short reflex enters through the enteric nerve, so this is the gut brain, right? The long reflex is through the central nervous system, will increase sympathetic, decrease parasympathetic, and basically will cause the reduction in contractile force. Now, once this happens, this can actually come and inhibit this presence after a time. So, and this, you know, we've just talked. So this is the entogastrons that can be secreted that'll affect, and this is the enterogastric reflex. I think I'm almost killing that to the point of being ridiculous. So homeostatic imbalances that can arise from the stomach. Vomiting is basically caused by extreme stretching. And so this is intestinal, right? Extreme stretching can cause this. Intestinal irritants such as bacterial toxins or excessive alcohol, spicy food, certain drugs can all cause vomiting. And basically what happens is chemical and sensory impulses stimulate the emmeric center in the medulla. So the problem with excessive vomiting, it can lead to dehydration and electrolyte and acid base. And so alkalosis, right? And so a lot of times we want to get that under control. So now let's go, and we've basically beat the stomach to death. Let's go and talk about some of the other things that work. So the liver, the gallbladder, and pancreas are accessory organs. They're not actually part of the tubing that goes through. So the liver is important because it has the digestive function, and a main part of it is for the production of bile. And bile is a fat emulsor. So it's not so much going to digest fat, but it's going to allow big clumps of fat to be put into smaller kind of droplets. The gallbladder, its chief function is the storage of bile. And the pancreas supplies most of the enzymes to digest chyme. It also is important because it produces most of the bicarbonate to neutralize stomach acid.
There's a factory inside you that weighs about 1.4 kilograms and runs for 24 hours a day. This is your liver, the heaviest organ in your body, and one of the most crucial. This industrious structure simultaneously acts as a storehouse, a manufacturing hub, and a processing plant. And each of these functions involves so many important subtasks that without the liver, our bodies would simply stop working. One of the liver's main functions is to filter the body's blood, which it receives in regular shipments from two sources. The hepatic artery delivers blood from the heart, while the hepatic portal vein brings it from the intestine. This double delivery fills the liver with nutrients that it then sorts, processes, and stores with the help of thousands of tiny internal processing plants known as lobules. Both blood flows also deliver the oxygen that the liver needs to function. The blood that is received from the intestine contains carbohydrates, fats, and vitamins and other nutrients dissolved in it from the food you've consumed. These must be processed in different ways. In the case of carbohydrates, the liver breaks them down and converts them into sugars for the body to use as energy when the filtered blood is sent back out. Sometimes the body has leftovers of nutrients that it doesn't immediately require. When that happens, the liver holds some back and stacks them in its storage facility. This facility works like a pantry for future cases when the body might be in need of nutrients. But the blood flowing into the liver isn't always full of good things. It also contains toxins and byproducts that the body can't use, and the liver monitors these strictly. When it spots a useless or toxic substance, it either converts it into a product that can't hurt the body, or isolates it and whisks it away, channeling it through the kidneys and intestine to be excreted. Of course, we wouldn't consider the liver a factory if it didn't also manufacture things. This organ makes everything from various blood plasma proteins that transport fatty acids and help form blood clots, to the cholesterol that helps the body create hormones. It also makes vitamin D and substances that help digestion. But one of its most vital products is bile. Like an eco-friendly treatment plant, the liver uses cells called hepatocytes to convert toxic waste products into this bitter greenish liquid. As it's produced, bile is funneled into a small container below the liver, called the gallbladder, before being trickled into the intestine to help break down fats, destroy microbes, and neutralize extra stomach acid. Bile also helps carry other toxins and byproducts from the liver out of the body. So as you can see, the liver is an extremely efficient industrial site, performing multiple tasks that support each other. But such a complex system needs to be kept running smoothly by keeping it healthy and not overloading it with more toxins than it can handle. This is one factory we simply can't afford to shut down.
So let's look at the gross anatomy of the liver really quick, and we're going to do this in class as well, right? This is the largest gland in the body. It weighs something about 3 pounds. Right? It consists of 4 lobes. You have the right lobe here that you can see very easily, and that is separated here. Actually, let me do, oops. This whole area down here is the right lobe. Then you can see the left lobe over here, and the two is separated by this falciform ligament that's in the middle. That's going to separate the right and the left lobes. Also, it suspends the liver from the diaphragm and the anterior abdominal wall. Now, if we're looking for the other lobes, you're going to have to flip around it, and you'll see that you can see here, here's the left lobe. Again, this is the right lobe over here, and you can see on the bottom here towards the back, you can see this part right here, which is the caudate lobe, and then the lobe that's between this falciform ligament and the gallbladder at the bottom is the quadrate lobe, right? Now, if you look here, you see the filiform ligament, and then a little farther, you see this round ligament. This is also known as the ligamentum teres. And this is the remnant of the fetal umbilical vein. Umbilical vein. So that's along the free edge of the falciform ligament. Let's see. You can see here, you have the gallbladder, which is going to hold the bile. You have, from there, you have the cystic duct that is connecting to the gallbladder. You can see this common hepatic duct. This is the duct that leaves the liver. And basically the bile duct, which you can see right here, is the connection of the cystic duct and the common hepatic duct together. Let me see, let me go back one. And so you can see here, okay. You can see on here as well, you have the hepatic veins here, and you have the hepatic artery here, and they basically enter the liver at this porta hepatis region. Now, when we look at the liver itself, this is a large anatomy. We can look at the microscopic anatomy, and when we look, this is the anatomy of the liver itself. And so on here, you can see we're taking the liver and we're taking these, lobules right here, and we're blowing them up, right? And so these guys are composed of basically hepatocytes. And so you can see that this lobule up here, right, you can see all those. So these little individual things here are all hepatocytes. And those cells help to filter and process nutrient-rich blood. You can see the central vein here. And that's located in this longitudinal axis. If you look in each corner of the lobe, it'll contain what's known as a portal triad. And the portal triad is made up of three things. The portal triad is made up of this, and you can see where the green dots are, the hepatic artery. This supplies O2. The hepatic portal vein, which brings the nutrient-rich blood from the intestines. And the bile duct up here, which is going to essentially receive bile. And it receives bile from these little guys right here, if you can see. It's the bile caniculus. Now, you also have these sinusoids that you can see here. These are leaky capillaries, and they're located between the hepatic plates. And so the blood from both the hepatic portal vein and the hepatic artery actually percolate through the triad region that I just showed you here, right? And from the triad region, they go through the sinusoid and empty into this central vein area here. And you can see on every single one of these, they have these portal, these triads. And you can see they're all moving into the middle here. So you can see again, just to review them, this whole thing, right, is the lobules. They're composed, again, of hepatic cells or hepatocytes. They help to filter and process nutrient-rich blood. You can see this central vein here. And so what happens, you have these triads that are in the corner, right? And if you remember, these arteries supply O2. The portal veins bring nutrient-rich blood from the intestines. The bile ducts receive bile. And then all of that is going to go and move through these sinusoids to empty into the central vein. Now, these bile caniculi here are going to be what ends up bringing the bile into the bile duct. And within this area, you have stellate macrophages, otherwise also known as hepatic. And these are in the liver sinusoids. And they help to remove debris and old red blood cells. So let's really quick talk about the hepatocytes. And so the hepatocytes have increased rough and smooth ER, Golgi apparatus, peroxisomes, and mitochondria. And they have the following functions. They help to produce about 900 milliliters of bile a day. They process Bloodborne nutrients. Like example, store glucose as glycogen and make plasma proteins. They store fat-soluble vitamins. And they detoxify. So, perfect example, they take something like ammonia and they convert it into urea. Now, when we're talking about hepatocytes making bile, what exactly is bile? And what it is actually this yellow-green alkaline solution, and it contains bile salts, bilirubin, and things like cholesterol, triglycerides, phospholipids, and electrolytes. And basically, the enterohepatic circulation helps to recycle bile to conserve bile salts. And so there are some homeostatic imbalances of the liver. Hepatitis, for instance. This is usually a viral infection or can happen due to drug toxicity or wild mushroom poisoning, or something like cirrhosis, which is a progressive chronic inflammation from chronic hepatitis or alcoholism. So basically what happens is the liver, those areas get converted into fatty, fibrous, useless cells. And this leads to what's known as portal hypertension. And so basically the liver isn't able to detoxify your body the way it should. And so a lot of times liver transplants are successful, but the problem is livers are scarce. Liver can regenerate to its full size in 6 to 12 months after 80% of it is removed, but you have to have some of it left, and that part needs to be somewhat healthy. So it can cause a bit of a problem. Now the gallbladder. This guy is a thin-walled muscular sac. It's on the ventral surface of the liver. And its function is to store and concentrate bile. And they do that by absorbing water and ions. It contains many honeycomb folds that allow it to expand as it fills. And basically, muscular contractions release bile via the cystic duct, which flows into the bile duct. You can get basically gallstones is a homeostatic imbalance. It's caused by too much cholesterol or too few bile salts. It can cause obstructions of flow of bile from the gallbladder. It's painful when the gallbladder contracts against sharp objects, crystallized objects. So this can be very painful. Obstructive jaundice is also an issue that can happen, and this is a blockage that causes bile salts and pigments to build up in the blood, resulting in jaundice or yellow skin. Jaundice can also be caused by liver failure. And basically, gallstone treatment is taking either crystalline dissolved drugs, ultrasound vibrations can allow movement, laser vaporization, or surgery. And now we're going to hit on what the pancreas is.
Beneath your ribs, you'll find, among other things, the pancreas, an organ that works a lot like a personal health coach. This organ controls your sugar levels and produces a special juice that releases the nutrients from your food to help keep you in the best possible shape. The pancreas sits just behind your stomach, an appropriate home, as one of its jobs is to break down the food you eat. It aids digestion by producing a special tonic made of water, sodium bicarbonate, and digestive enzymes. Sodium bicarbonate neutralizes the stomach's natural acidity, so these digestive enzymes can perform their jobs. Lipase breaks down fatty substances, protease splits up proteins, and amylase divides carbohydrates to create energy-rich sugars. Most of those nutrients then get absorbed into the bloodstream and go on to enrich the body. While all this is happening, the pancreas works on another critical task, controlling the amount of sugar in your blood. It achieves this with the hormones insulin and glucagon, which are produced in special cells called the islets of Langerhans. Having too much or too little sugar can be life-threatening, so the pancreas must stay on constant alert. After a big meal, the blood often becomes flushed with sugar. To bring us back to normal, the pancreas releases insulin, which makes the excess sugar move into cells where it's either used as an energy source or stored for later. Insulin also tells the liver to shut down sugar production. On the other hand, if blood sugar is low, the pancreas releases a hormone called glucagon that tells the body's cells and liver to release stored sugars back into the bloodstream. The interplay between insulin and glucagon is what keeps our sugar levels balanced. But a faulty pancreas can no longer coach us like this, meaning that this healthy balance is destroyed. If it's weakened by disease, the organ's ability to produce insulin may be reduced or even extinguished, which can trigger the condition known as diabetes. Without regular insulin release, sugar steadily builds up in the blood, eventually hardening the blood vessels and causing heart attacks, kidney failure, and strokes. The same lack of insulin deprives cells of the energy-rich sugar they need to grow and function. People with diabetes also tend to have higher levels of glucagon, which makes even more sugar circulate. Without this internal health coach, our sugar levels would go haywire, and we wouldn't be able to digest important nutrients. But like any coach, it's not the pancreas's job alone to keep us healthy. It needs our conscious participation too.
So I hope that helps a little bit. So let's talk about the pancreas. It has exocrine and endocrine function. So if you're looking at the cell here, you'll see a small duct, and you can see basically you have the acne, which is a cluster of secretory cells. They produce zymogen granules, which actually contain what's known as proenzyme. Now you can see on there that you also have ducts. And so these guys are going to secrete to the duodenum via the main pancreatic duct. Smaller duct cells can produce water and bicarbonate. So you can see the center cells that are there to help basically secrete enzymes, and you can see these zymogen granules that are in there that are basically the proenzymes before they're actually activated. And then you can see that that is incorporated when one of these sinus, and then the small ducts go into here, and these are going to actually secrete to the duodenum via the pancreatic duct. So this is the exocrine function. Now they do have an endocrine function because it secretes both insulin and glucagon. And these are made by the pancreatic islet cells. And so you can see these small duct cells that create the bicarbonate and water are there as well. And these are the larger ones that help to secrete the actual enzymes that we're going to use to digest. And so about 1,200 to 1,500 mils per day of pancreatic juices are produced. It's a watery alkaline solution, about a pH of 8. And essentially, it's there to neutralize the acid chyme coming from the stomach. Wait a second, I'm trying to figure out. And so basically, you have the electrolytes are produced primarily, but then you also have digestive enzymes. And so the digestive enzymes that we have are things like proteases. And so these are for proteins. And they are released in an inactive form. You don't want to have self-digestion. You have amylases, which are for carbs. You have lipases, which are for lipids. And then you have nucleases, which are for nucleic acids. And so the pancreatic juices, the proteases are secreted in an active form. They are activated after they reach the duodenum. And basically they're activated by this enteropeptidase, right? And so this guy is going to interact or activate trypsinogen to trypsin. And once trypsin is activated, it can then activate more trypsinogen. And then you also have things, no, like procarboxypeptidase. Gets activated into, you just take the pro into carboxypeptidase, and then you have basically chymotrypsinogen that gets activated to basically chymotrypsin. And you can see them all over here. I realize that they were over there. And so these are all the inactive forms, and basically you can see as trypsin gets activated, it helps to activate these other. And so these are all examples of proteases. They're all going to break down different proteins or types of proteins. And so you can see how everything's kind of tied together here, right? You have the liver here, and you can see that you have the hepatic ducts here and here. Here's your gallbladder, and they all go into, and you can see the bile duct here that kind of goes, and it all kind of combines over here together, where you have this main pancreatic duct and sphincter head together and release here in this major duodenal papilla area here of the duodenum. And so the bulb-like area is what's known as the hepatopancreatic ampulla, and what closes it and opens it is the sphincter. And so this guy controls how much bile and pancreatic juices get in there. And this accessory duct that's right here dumps directly into the duodenum. It's much smaller. And so, mechanisms that promote secretion and release of bile and pancreatic juices. So bile and the pancreatic juice secretions are both stimulated by neuronal and hormonal controls. And so the hormonal controls are just like we were talking about earlier. Oops, sorry guys. Wait. You have the CCK and secretin that are secreted by the duodenal enteroendocrine cells. And so the CCK is stimulated by protein and fat in the chyme. The secretin is released due to acidic chyme. And basically, these two guys enter the blood circulation and cause the following events to happen. First, it causes pancreatic secretion, right? And so you can see this is going into the pancreas. And so the CCK is going to induces secretion by the center cells of the enzymatic pancreatic juices. And the secretin causes secretion by duct cells of the bicarbonate-rich pancreatic juices. And so the vagus nerve is weakly stimulated during the cephalic and gastric phase to cause change here too. Now from here, you can see that the CCK and secretin activates the liver to start secreting bile. And the bile salts returned from the interhepatic circulation are the most powerful stimulus for bile secretion. Secretin is such a minor stimulus that it's not as important. So here, it's all about the bile salts, right? After that, gallbladder contracts, because that's what's holding the bile, right? And CCK has it to contract. The vagus nerve does have some workings in this, but it's really weak. And so you can see here, the hepatopancreatic sphincter is going to relax. And CCK causes that sphincter to relax. And so at that point, bile and pancreatic juice actually enters the duodenum. So CCK and secretin are important throughout this. Both of them are important in this first part when we're talking about the pancreatic secretions actually happening. Once that happens, basically bile, you'll see some of this CCK coming into the liver is more important here. But for bile secretion, the most powerful part of it is the returning of bile salts, right? And then you'll see the gallbladder contracts to release it due to CCK. And then basically the sphincter will relax because of CCK. So the only time secretin is a really big factor is the very first part when we're talking about the cells actually secreting it to talk to the pancreas. So secretin's really important in pancreatic juices, and the rest of the area it's not so much. And the small intestines length and other structural modifications provide huge surface area for nutrient absorption. So that's what's important here. And so there are some modifications. And so if you look at here, you'll notice on there that you have these microvilli that are part of this brush border cells that help with surface area. So you can see there's so much more area that's exposed. So that's going to help the basically the absorption of nutrients. Missing. And so when we're talking about the digestive process in the small intestines, the chyme from the stomach actually contains partially digested carbohydrates and proteins and undigested fats. Hold on here really quick. And so when we're talking about how long it takes the small intestine to absorb all the nutrients and most of the water, on average, 50% of the stomach content empties about 2.5 to 3 hours and completely empties about 4 to 5 hours. So it requires about 2.5 hours to 3 hours before 50% of the content in the small intestines actually emptied. And at this point, your digestive system has absorbed most of the nutrients, right? Sorry. So sources of enzyme for digestion, substances such as bile, bicarbonates, digestive enzymes, not brush border enzymes, are imported from the liver and pancreas. Brush border enzymes found in the plasma membrane perform final digestion of chyme. And so the process in the small intestines, how it moves, after a meal is segmentation to break everything up, but between meals, it's peristalsis to make sure to get rid of everything. And then the last part of this is the large intestines, and this is the area where we're just going to basically absorb water and get rid of what we've digested. And you can see in here, it's made up of 3 parts: the ascending, transverse, and descending colon along with the sigmoid colon at the end. Towards the bottom, you'll get the rectum and the anal canal with both external anal sphincter and then internal anal sphincter. You can see the mesenteric here as well. And so, and of course, the very first, as you can see, this is the ileum. That comes from the last part. So you have the duodenum, the jejunum, and the ileum of the small intestines. And so the ileum comes out to enter this first area called the cecum into the small large intestines. And then it'll start coming up from there. Of course, the appendix, where some people, it's still kind of unknown completely what it does. It aids in some digestion.